Hello everyone, my name is Rahul. In the previous video, we had discussed about downstream processing. In this video, we are going to start with the applications of biotechnology. Firstly, we are going to discuss application of biotechnology in agriculture. Now, to increase crop production, we have largely three methods. First is agrochemical based agriculture. Next, we have organic agriculture. And the other is the use of genetically modified crops or genetically modified organisms, which are also known as GMOs. Now, first we will start with the Green Revolution. Green Revolution was primarily dependent on agrochemical based and organic agriculture. So we will not be going into much detail. The very details of this Green Revolution you will study in the chapter on strategies for enhancement in food production. So Green Revolution refers to the period in mid 1960s when the crop production of India increased and almost tripled from its original value. So, Green Revolution is the period in the mid 1960s when we became from a country of crop deficit to a country of crop excess. Now, Norman E. Borlaug is known as the father of Green Revolution. He is known as the father of Green Revolution in world. And Dr. M.S. Swaminathan, he is the father of Green Revolution in India. Now, Green Revolution was primarily dependent on agrochemical based agriculture and organic agriculture. Now, why are we studying this in biotechnology? Because the Green Revolution, it used a primitive method of biotechnology which basically dependent on cross fertilizing between two plants whose characteristics were desired. So let us say there was a plant which had a very long life and a plant which had a very good produce. So what we would do is we would cross these plants together and in the hope that the progeny will incorporate both the characteristics of long life and good produce. However, the disadvantage was that only 1 out of 1000 or 10,000 cross fertilization would actually result into a progeny that we needed. So later on, we shifted on to biotechnological processes when we would just take up the gene which was responsible for good produce and incorporate into the plant with a long life. Nevertheless, Green Revolution was primarily dependent on the use of primitive type of biotechnological methods. So we will see the crops which were a result of a primitive type of biotechnology. So in 1963, the year 1963, there were two varieties of wheat, namely Sonora 64 and Lerma Rojo 64, along with a Japanese variety called as Norin 10 were introduced in India. Now, these varieties they had very good produce, but they could not adapt to the Indian conditions. So, these varieties they were subjected to mutations. Mutation means a change in the genetic material, subjected to mutations, and they were finally able to adapt to the Indian agriculture, Indian conditions, subjected to mutations and artificial selection Artificial selection means selecting only those progenies, only those mutated progenies which had our desirable characteristics, and they could finally adapt to Indian system, Indian conditions, and provided us very good produce. This was done at the Agricultural Research Institute, Indian Agricultural Research Institute, New Delhi. So this is a primitive form of biotechnology when we are mutating the entire genome of the plant rather than selecting one particular gene and incorporating it into a new plant. Again, there were other varieties, namely Sonalika and Kalyan Sona. These were very high yielding varieties, high yielding, disease resistant varieties of wheat. They were also introduced in the wheat growing belt of India and they provided very good results. These varieties, Sonalika and Kalyan Sona, were essentially derived from the varieties mentioned above, that is Sonara 64, Larma Rojo, and Norin 10. So during the period of 1960 to 2000, wheat production increased from 11 million tons to 75 million tons. Similarly, there was improvement in the quality of rice. How come? So in the rice, what we did was there was a gene known as dwarfing gene called as DGO Wu gene. A dwarfing gene called as DGO Wu gene was noticed in Taiwan. This gene produced many improved varieties of rice. So this gene was picked up by IRRI, which stands for International Rice Research Institute, which is situated in Philippines. So this gene was picked up by IRRI and introduced into the rice varieties, so which resulted in production of high yielding varieties IR8 and IR24. So these varieties were developed from this dwarfing gene. So what an Indian scientist did was he crossed 13 varieties of rice taken from 6 countries in wild rice to develop IR36 variety of rice. So an Indian scientist who was named as Gurdev S. Khush, he crossed 13 varieties of rice with Indian wild rice and he produced a variety known as IR36. IR36 was a resistant variety which was resistant to grassy stunt virus. It was also a high yielding variety of rice and solved a major food problem in India and in the entire Asia. Later on, after IR6, later improved high yielding varieties called as Jaya and Ratna, they were introduced in India. They were developed and introduced in India. So again, during the period from 1960 to 2000, rice production increased from 35 million tons to around 89.5 million tons. Now, there is plant breeding for development against resistance to insect pests. Now this we will take up in the next video. Before that, we will do plant breeding by mutation. Plant breeding by mutation. Mutation means changing the genetic material to provide or to change the characteristic of an individual plant. So what we did was we mutated some of the plants, we played with their genes, mutated them. Rather than selecting one particular gene, we took up the entire genome, mutated the genome in a search that one particular gene of interest will be mutated and provide us the required plant variety. So an example of this is a variety of Abolmuscus esculentus, commonly known as bhindi, resistant to yellow mosaic virus, was developed from a wild variety which was naturally which was naturally resistant to this disease. This variety is known as Parbhani Kranti. So apart from wheat and rice, other variety of bhindi which was developed from a wild variety is known as Parbhani Kranti. This is also an example of plant breeding by mutation. Now plant breeding for disease resistance you will study in the chapter on strategies for enhancement in food production. With this we finish off with this video. See you in next video.